The overall goal of this novel imaging setup and operating procedure is to perform photoacoustic microscopy and optical coherence tomography dual modality for non-invasive, label-free, chorioretinal imaging of the eyes of larger mammals, such as rabbits.
This method can help answer key questions in ophthalmic imaging about the imaging depth, resolution, and clinical translation required to discern oxygen saturation, blood distribution, and melanin in larger animals.
The main advantage of this technique is that it allows non-invasive visualization of ocular tissue for the acquisition of photoacoustic microscopy and optical coherence tomography dual-modality images.
To begin, select an optical parametric oscillator laser pumped by a diode pump solid-state laser as the light source for the photoacoustic microscope and use a beam splitter to divide the collimated beam with a split ratio of 90 reflection to 10 transmission. Successively deflect the reflected portion by the M3 mirror in a dichroic mirror and use a two-dimensional galvanometer to raster scan the reflection. Deliver the scanned beam through a telescope composed of a scan lens with a focal length of 36 mm and an ophthalmic lens with a focal length of 10 mm and focus the beam on the fundus by the rabbit eye optics. Before beginning the imaging procedure, place a water circulating heating blanket on the body support and set